Dr. Armelle Pindon. She is our Director of Scientific Engagement and Collaboration, and she has 20 years of experience working in academic research in the pharmaceutical industry, and she is now working for us. So, Armelle, would you like to come up?
Okay, so now I think you understand everything about VCP. So I'm not going to talk about VCP. I'm going to talk about something very boring. It's drug discovery. I need the thing. So I'm going to try to make you understand the reason why we choose our strategy. So let's begin with the basics. So what are the aims of Cure VCP Disease organization? It supports the patients, and Nathan and Alison do a fantastic job on that, and find the treatments, and that would be my priority. I love you all, and I'm going to support you as much as I can, but my role really in the foundation is to find the treatment. So what can we do? We can become a support for drug discovery and drug development. That does not mean that we are not going to support and collaborate and trigger basic science. I think Dr. Well just told you that we have this huge program that we hope will be funded on FTD and phenotype, and I have also triggered some collaboration between Alisa Johnson on lysosome with other groups in Italy. So it's not true that we are not going to do that, but for now, I'm going to explain you how we choose scientific projects that are crucial to drug discovery. So we say drug discovery. What's that, drug discovery? It's a process in 2 steps. First, you have to discover the drug, so that's drug discovery. And when you think you have a drug, then you need to develop it. So that's the preclinical part, you look for the drug, and the clinical part, you are going to go in patients and develop the drug. So everything starts with investigating the characteristics of the disease, so understand the problem. What's the problem in the patients? Then you need, I have this thing just in my eyes. You find a chemical compound, so what's that? What does that mean? So you find a way to solve the problem that you just identified. So you find a chemical compound, you test it. When you see it works, you go in animal models that Chris just explained, and then the chemists are going to enter in the field and modify the molecule that we call an optimization, so that it becomes really a drug, a chemical compound that is going to be good enough to be a drug. And then you go to humans. So you go to patients, you test the drug in humans, and then you go on the market, and then you get the feedback from the large population. So how does it work? Really, if you see globally, so I just explained that, who does that and where? The first part is done by biologists in academic science. The second part is usually done by pharmacologists, biologists, medicinal chemists, and ADME toxicologists. That's a big word. So toxicologists, you can imagine, they are going to check for the toxicity. Is the chemical compounds we have, the future drug, is it going to kill you if I give it to you? So that's the toxicology, or not kill you, but maybe you will become blind, or that's the toxic part. ADME means how your body is going to absorb it, distribute it. Is it going to go to the muscles, to the brain, to the gut? And the E is for extraction. So how does your body is going to reject it? So there is a whole process in drug discovery, and you cannot improvise this kind of process. It has really to be done by experts. So when you're sure that your drug is going to be good enough to go in patients, the clinicians in hospitals are going to test the drug. And then the pharma is very happy and it goes in business development. So why do you think I presented that just for you? Because you can act on every of these single, sorry for the errors. This is the patient advocacy group, and you can act and be involved in every step of the process. The other institutions that are involved are the FDA, of course, who are going to decide, look at your results at the preclinical steps and say, okay, is it good enough to go to human? Is it safe enough to go to human? And they will take the decision to go there. Then, when we have enough results on the humans, they are going to, okay, is it going to really be beneficial for the patient? Does it really make a difference in their life? Balancing with the toxicology, because everything at a point is toxic. And then they make the decision to go to the market. Who is intervening in that? It's also the pharma and the biotech, the people who are going to pay for it and get money from it at the end. So they are going to intervene in how to find the chemical compounds. Then in the clinicians, they are going to pay, that costs millions of dollars, and finally they will go on the market with the business development. So going back to the beginning, how do we start as patient association? Oh, doesn't work. Oh. Okay, so you want to find where to act. And as Brandon said, I spend a lot of time in pharmaceutical industry, and my boss would always say, okay, I would come to my boss and say, oh, I have a new target. I know what I want to do. And he said, okay, is it smart? You need to be smart to be druggable. So is it specific? Can you make an alteration of one element or everything, and you don't know what you are acting on? Is it measurable? Do you have a way to evaluate the alteration? Is it assignable? Do you have the expertise in-house? Do you have an expert who knows how to do it? It has to be realistic, doable. It's not that you are going to investigate for 5 years before you can start your project. So it has to be something with not a big challenge. And it has to be time-bound. You can measure the effect in a short time. As Chris mentioned, if you need to wait 2 years before you can measure anything, that's not a good target. So that's the first thing we evaluate to find a target. So that's the first step is target identification, where you can intervene to fix the problem. So if it's smart, then you are going to go to target validation. If I modulate that target, is it really going to change the manifestation of the disease in assays of reference? So what does that mean? So, for example, Vcp inhibitors. You all heard about the fact that there are Vcp inhibitors that were developed for oncology. Are they tested in several cellular models? Are they tested at different doses in the conditions that are relevant for VCP disease? So that's something we need to develop. We need to agree on the tools for validations, because now for VCP inhibitors, for example, we have the tools for validation for oncology, but the tools for validation for the VCP disease is still to be developed. For example, you have some scientists that would say, okay, the VCP with the mutation produces a lot of energy, so we need to calm down, what we call inhibit. But others would say, well, it produces a lot of energy, but the function of VCP is to take that energy and move, get this movement to get the protein through. So what is the right assay? Is it the amount of energy that the VCP is going to produce, or is it the function of taking the protein and going up and down? So we really need to agree on the assays and agree on the models relevant to VCP. If you take different examples, for example, if you measure, can you drive? Okay, Are you the fastest driver? If you measure on a nice road for 100 miles, or if you measure for half a mile in mountains, or if you measure with those poor guys who try to just go ahead, well, you can answer, that's the same question, but which test is relevant to your question? Okay, be careful. I'm going to start really with science now. Breathe, Nathan. I'm beginning very slow, so with the basics. So imagine you want to make some food. So you go to a cookbook. You open the cookbook. There is the picture of the guy who wrote the cookbook. You have nice pictures of anything, the region, Tuscany, I just come back from there. But you have also the recipes. And all the recipe, all the items in the book that are relevant if you want to make food. From those, you will read the recipe and finally have a product after reading the recipe and doing what the recipe asks. So just remember that. Okay. So in cells, if you think about the cookbook, you have the DNA, which has all the information. In that DNA, you have images and biographies that will not help you creating something. And you have other parts of the DNA that are genes. And those genes are the recipe. And the whole different genes that you have in the DNA is called the genome. From those recipes, you are going to make RNA and read the RNA, the instruction, to produce a protein. So remember, you have the gene, the DNA, then you have the RNA, and then you have the protein, which is the end results. Am I clear? Yeah? Okay. So now, thinking about the targets, where do I want to act to find a treatment? First, you can act at the origin of the problem. What is it for VCP? It's a gene mutation. So what would be the target? That would be the gene or the gene transcript. The gene transcript is the RNA that I just mentioned. So you can target that. You can say, okay, I'm going to modulate that. You need validation. If you say, that will be my target, you need a way to measure if I remove the gene that is bad. I need a way to measure, is it going to be better or not? So that's, it goes without, did I push it? So you can go to the next level. It goes by itself, that's magic. The consequence of the problem, that would be the protein. Whoa, I'm missing something? No, okay, how can I go back? Sorry for that. So the consequence of the problem is that the protein, the VCP, doesn't work. It doesn't do its function. So what would be the target in that case? That would be, no, I'm going the wrong direction. That would be the protein in itself. So you can either replace it, you purify protein and you give it to the patient, or you can modulate it, and then you need a modulator. The indirect consequence of the gene mutation is that the protein VCP doesn't work alone. Most proteins don't work alone. They have co-workers, what we call for the VCP cofactors. So, and there are consequences, because if the VCP cannot work, then also the cofactors, the co-worker, will not be able to do their job. So that's what we call the downstream signaling pathway. And that can be also a target, like the TDP43 that was mentioned several times before. That can be also a target, but that would be really at the end of the chain. So when you have your target, then you need a chemical compound that you are going to test and bring it to the drug. So you need chemical libraries that you are going to put on the biological target. So there are different molecules. You look at the one that is going to stick to your protein and modify its activity. And then you tell the chemist, I got a hit. Then it takes the hit, modify it, add some parts, remove some parts, and try to identify which part of the molecule, if you modify it, is going to be better, stick better, activate more, inhibit more, modulate, how it's going to modulate your protein. And that's what we call the structure-activity relationship. That is extremely important in chemical compounds. So after that, you have a really nice molecule, effective, and that we want to call it a drug. So where can we do that? Mostly in biotech and pharma, but now we have also the NCATs, which is from the NIH, where they are really specialized in drug screening. And you have specific institutes where you have people that are really doing only that, experts. What do we use? We can use general chemical libraries, so a bit everything, drugs that can act on, I don't know, headache, and you try it on VCP and you see if it works. Or you can have specialized libraries where you have some compounds that you know that are going to work mainly on neurons, other that are more specialized in muscles. So that's the two different approaches. So another way to, another very important step is in what you are going to test. So you need to know what activity you want to measure, but you need to know in a representative of the disease. So you can go from very general, and you have what we call a house throughput readout. You can do thousands of compounds at the same time. You can go in yeast, you can go in animal cells that you have genetically modified. Why is it going by itself? You can have humans, genetically modified or from patients, and then you can go to the different model we had. So we have Drosophila, which is a fly. We have zebrafish that were genetically modified to have the VCP mutation. We have, and I would like to develop that a lot more, patient-derived 2D tissues and 3D organochips models. So the FDA is really focusing on that now, which also push us in that direction, which is way more relevant to the disease because it's human tissue and easier to work than mice. So we have different mice model, and Chris has developed 3, I think, different mice models. Do we need vertebrate mammal non-rodent model like mini pigs? The FDA loves it, but it's very expensive. So we know what we want to test. We know that we need to, and shouldn't push and do that. We have models, but they could be better. They could be more relevant to the disease in patients. So that's something we need to work on. So now you know the traditional target-based drug discovery process starts with target identification, then goes to validation, screening. You're going to identify some molecules, and then you improve the molecules, and you go to patients. It's a very long process. It takes around 10 years and even sometimes 15 years to do that. So where are we with VCP? We have identified some targets. We have started, some labs have started to screen the molecules, but we don't have good target validation. For the inhibitors, for example, they were tested in, sorry, oops. So they were tested in mouse model, but we don't really know, as Chris explained, we don't really know, do we need to inhibit the VCP or do we need to activate the VCP, how to modulate it? We don't know exactly, so we need to validate that first. So if we want to save time, something that is very important is repurposing drugs. So what does that mean? So you have the whole process, but you have drugs that already exist, like the drugs with Amylix on the autophagy and Cleave Therapeutics. So we can put them back in our model for VCP disease, check it, and go to human. That means that you skip a whole process, you reduce time, and you reduce cost. So you go faster. So that's one of the strategy we have at the organization. Another way to screen is not with small molecules and target identification. Another way is to go through phenotypic screening, so that using iPSC. So that will be the most difficult part. Sorry, and after you can relax. So when you are at the very beginning of the embryology, you are basically a bag of cells, a bag with a kind of group of cells, and there is no difference between one cell to another. You don't see an arm, you don't see a foot, you don't see muscles, anything. They are what we call stem cells. And all those cells during the embryology, the development, are going to differentiate. They are going to be different one from the other. One is going to be a muscle, the other one is going to be a neuron, the other one is going to be a bone. So that's what we call the differentiation. And I wish I would be part of the we. Scientists have identified small molecules that can trigger this differentiation. So you take the stem cell, you put some specific molecules on it, and it's going to become, depending on the molecule you put on it, a muscle, a neuron, a bone, whatever. The beauty of it is that we also identified a way to go backward. So you take a cell that is already differentiated, for example, from your skin, you put some molecules on it, and you are going to go back to this kind of cell. So we take your cell from the skin, we put it back to embryonic, and then we put back some molecules, and we are going to create your muscle, your neurons, and we can test drugs on really your muscles and your neurons, not just a muscle or a neuron. That's what we call the iPSC. So we can put, for example, if we decide we take your skins, we put it back to stem cell, then we decide, okay, we are going to study your muscles. So we differentiate them in muscles, and then what can we do? We can put all those cells in plates where you have 96 wells, 96 possibility, or 384 possibilities, and we are going to look at the contraction force of your muscles, the mitochondrial activity, the energy, that's the energy, the morphology of the muscles. Ooh, go back. The morphology of the muscle. So we can look at many different things, many different characteristics, put drugs on it, test maybe 100 different drugs on your muscles, and look if we have an action. So the process is totally different from the process I was explaining at the beginning. We put a lot, like thousands of compounds on your muscles, and then we have a result, and then we need to do what we call target deconvolution. So we try to go back and find the target. So it's a totally different way from the beginning. When you have a target, you screen the compounds. Now you screen the compounds and you say, oh, but what was the target? So why do we do that? First, when you want to have money and you want to have the compounds, you go to pharmaceutical industry and you tell them that you are a very small rare disease community and you have a very poor understanding of the disease. You don't know exactly what phenotype is associated with which variants, and it's very confusing. It's complex mechanisms, so they don't like it. Then you have expensive failures, usually with the very traditional target identification. So with the iPSC phenotype screening, you reduce that risk. Sorry, it goes by itself. So the phenotypic screening is adapted for more complex mechanisms, such as the VCP disease. So I hope I didn't lose you. So I'm going to go to gene therapy, which is simpler. So there are two types of gene therapy that we can do with VCP. Is either what we call gene replacement, but it's in fact adding the gene to the cells, hoping that's, but the variant gene is still there. So hoping that you put enough so that it will compensate the fact that you have a variant. The only problem with VCP is that if you put too much, you have a risk to trigger different type of cancer. So that's a tricky measurement, and be careful not to put too much and not to put too little. And we don't know if we add some genes that are wild type, so no variant. As Chris explains, there's an hexamer. So it means that there are 6 VCP next to each other, making a circle. There's a hole. So if you have one variant still, even if you put so many other that are wild type, no variation with the gene replacement therapy, and you still have one among the 6 that has the variation, is it going to be still okay? Is it going to function or not? So that's something we don't know. Another approach for gene therapy is the gene editing. So gene editing is, in fact, you are correcting the error. So that's a technique that is done with the CRISPR-Cas that so many of you are asking about. So the CRISPR-Cas is going to locate the mutation, the variation in the DNA, cut it, and with a modified type of Cas, replace it and put back the right sequence that you should have in the DNA. So is it going to work? One more time? No, they're doing it. So I'm gonna rest for a minute.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
So should I press to go to the next or? Yeah. Okay, so I want you to focus on the fact that they say that might be used for treatments. The problem with that, the first problem with CRISPR-Cas is that you need to bring it to the cell of the patients, which means the best way we have found up to now is to put it in a virus, and the virus is going to infect your cells. The problem is that in a virus, which the most used is AAV, you can put a certain amount of DNA. So you can put a certain size, and the CRISPR, just the CRISPR, I'm not talking about Cas, is already huge. So you need one virus to bring the CRISPR, but then you need another virus to bring the Cas. So which means that you are going to do 2 infections in the patient to bring the CRISPR and the Cas. The other thing is that it's quite toxic because you try to have the RNA that are going to find your mutation, but the mutation is only 1 nucleotide, so that's extremely difficult. Even if it's bigger, it's already difficult. So you don't know where the CRISPR and the Cas are going to. They are going to bind to your mutation, of course, but they are going to bind also to other sites, what we call the off-target. And they are going to cut pieces of your DNA. So that's very toxic. You imagine if it's a crucial gene that you are going to cut randomly like that. So we started. We should start in September in cells, and meanwhile we start with that. We hope that there will be other. I'm sure the research is so there's thousands of people working on that, so the research will go ahead and will address all these toxicity issues and delivery system, so that we can finally use gene editing therapy. So now, let's see that you have understand how to choose a target. You have understand the whole process from DNA to protein, I hope. Let's see, what did we choose as a strategy in VCP disease for the next year or more? So first, we decided to act on the gene. So by the gene editing therapy that I just explained you, that's going to be done in France at the Geneton with Ganesh Warti. It's not only gene editing. There will be a whole collaboration with clinicians, taking patients' biopsy, muscles biopsy. There will be immortalization of muscles, the bioblast. There will be a biobank, iPSCs created from the patients, and those are going to be used for gene editing therapy. All the material will be with our property from the organization, from VCP Cure VCP Disease. Do I work here? So that's when a pharmaceutical industry comes to me and asks me, do you have an iPSC? Do you have a model? Is it intellectual property free? Can you give it to me and I don't have to go through the whole university, and that has a whole problem. So we will have the intellectual property of those myoblast iPSC cells so that we can give it to researcher of our choice, but we can also give it to biotechs and pharmaceutical company. Gene replacement therapy is at the very, very beginning with Virginia Kimonis in UC Irvine. They are just looking at where the virus is going to infect which cells. The ASO, it's the antisense oligonucleotide, is done by a company called Ionis, and I was talking to Chris maybe to do a collaboration and try to test them. It's also at the very beginning. That's a way to suppress the allele that has the variant, hoping that the other allele is going to do the work. So that's the way we act on the gene, the strategy for the gene. Now about the protein, so we can think about replacing the VCP, so isolating the VCP protein and infuse it to patients. That's called enzyme replacement therapy. We have been working intensely with Cleve Therapeutics for Vcp inhibitors. So they have developed inhibitors for oncology purpose. They are at the end of phase 1. So they assess the toxicology, and the toxicology is good. It means that it's safe on humans. Now they want to go for the VCP disease in the neuromuscular field instead of oncology, but they don't have the money. So we work together to prepare everything so that we would be sexy for the big pharma, which means that we try to convince them that we are bigger than we are, because we don't know, maybe CMT patients have VCP mutation. The CMT2Y might be all hidden VCP patients, and we can join with Huntington. And so we prepared everything, and we said we will have a preclinical toolbox. So we will have all the right models, all the right assays, and you will be just take us, and we'll put the compounds directly in the right model with the right toolbox. So you don't have, we have no risk. We have the patients, we have everything. Just we are very sexy, just come with us. So we can act also on the whole body, like the phenotypic screening I explained you. There is another part in collaboration with an association which is called CMTA. They have patients with VCP mutation. We might be doing animal model or mouse models and work with Psychogenics, which is a biotech company. They have a new technology that is called a cube, where they put the mice that were treated with compounds, and they measure at the same time more than hundreds of different characteristics, the way they work, the way they remember. So they test 100 characteristics at the same time. So the throughput of the screening is really nice. So we can act, if you remember the different level you can act on to do target identification. At the bottom was the other, all the co-workers, the cofactors, the consequence, the last consequence of the mutation. So we talked with a company called DuPont Therapeutics that work on TDP-43 condensates, and they are trying to develop new medicine to remove those TDP-43 condensates. So we work with them. We should have talked to Amylyx in July, but they are in their FDA approval and everything. So we'll talk with them in September about autophagy regulation, about compounds they have, if they can include. They have it on for patient for ALS, and we are going to talk with them to include VCP patient to see if it's possible. One of the other consequences is a stress granule regulation for VCP. So we have Stephanie Moon in U Michigan, who received money from CZI to develop the assays Yes. And I'm looking to find some chemical libraries to start the screening, and also using the iPSC that wouldn't have IP, so that we can really do a screening just for us. The intracellular clearance, so we can also start screening for VCP cofactor. So that program will be for the future. It's not ready, but that's something we have in head. So that's just for the therapy, and we have the other parts of more understanding really how VCP disease works, which is with other collaboration. So we need, when we have a drug, that's great. Okay, we are going to find a drug. We need to measure it in humans. So for that, we need the natural history study. You are going to hear a lot about it. We need biomarkers. The FDA is going to say, what are the biomarkers? How can we measure if this drug, this new drug, is really modifying your disease? So that's a very important program, and I really encourage you to give your blood for that. So the screening program, we need the preclinical toolbox, so the assays and the cellular model, the animal models, really relevant to VCP, and we need to find chemical libraries. For the basic science, I just told you there is the VCP cofactor characterization program that we want to do, and the structure-activity relationship of VCP variants. So really look at when you have a variation in the gene of VCP, how is it going to affect the way the protein is going to act? More production of more energy, less movements. The motor is broken, as Will said. So Chris said, sorry. So that's the way. So what we can, to get started, it's critical that basic science and clinical research share their tools. And we need funding. We really need to have all the community starting to help us. We need money for the natural history study, for the biomarkers. Well, except if you get the money from NIH, that would be nice. The preclinical toolbox, and maybe the VCP cofactor program. So really, this is all in the aim that the pharmaceutical industry and the biotechs will come to us and say, okay, you're ready, you have everything done. I'm the last time. So we need to show that we can prove the concept and that we can check the drug for its efficacy. So that's the whole list well, I don't want you to read it, but the whole list we are working on for the preclinical toolbox. And I'm sorry if I lost you during all that talk. Please ask questions, read the glossary. You have just a very small extract of the glossary. Alison and I spend a lot of energy on it, and I hope it will help you. And you can thank Alison. She did an amazing job on that, and you wouldn't have the glossary without her, I can tell you. And thank you for being here.
All right. Thank you, Armelle. Yeah, we don't have time for questions, but we do have an expert panel coming up at the end today after Dr. Ghoshal. And